Today, 20th Jan 2022, MLT second year, subject is microbiology. Yesterday, we are discussing harmful effects of fungi. Today, we are discussed to beneficial effects of fungi. Beneficial effects of fungi. So, what is the benefits in fungi? First is decomposition, nutrient and carbon recycling. Nutrient and carbon recycling. Second point, biosynthetic factories. Biosynthetic factories, the fermentation property is used for the industrial production of alcohols, fats and citric acid, oxalic and gluconic acids. Third one is important sources of antibiotics such as penicillin. And fourth point is model organisms for biochemical and genetic studies. Example, Neurospora crassa. So, Saccharomyces cerevisiae extensively used recombinant DNA in technology, hepatitis B vaccine. Some fungi especially used for Saccharomyces cerevisiae, especially this type of fungi used in recombinant DNA technology. Means it is a genetics. Genetics consist of RNA and DNA. How to formation of RNA, how to formation of DNA, how to synthesis of RNA and DNA. So, is this type of Saccharomyces cerevisiae, this type of fungi especially used for DNA technology. Last point, sixth point is some fungi are mushrooms. Some fungi are especially in mushrooms. Seventh point, yeast provide nutritional supplements such as vitamins and cofactors. Some fungi like yeast, some yeast like fungi used for supplements of vitamins and cofactors. Vitamins are very important vitamins are very important in the human health. Why? Because without any vitamin deficiency, you have to face to the type of diseases, clear. Penicillium is used flavor of roquefort and combat cheeses. So, this ergot produced by Claviceps papyro contains medical importance alkaloids. Fungi, Leptolingia cordata apheromycosis is used to mosquito larva paddy. Especially this type of fungi is used for malaria purposes. Then next heading, classification of fungi. Depending on cell morphology, fungi classified by depending on their form morphology are four classes. One is yeast, second one is yeast-like fungi, third one molds, and dimorphic fungi. Yeast are unicellular fungi. Yeast are unicellular, only single cell. Unicellular fungi is known as yeast. Very important in Viva question. They are spherical in shape. Yeast are spherical shape. Spherical means is a round shape. And reproduce simple budding on culture. They form smooth creamy colonies. Yeast are on separates dextrose agar media. You have to see after growing is a creamy color colonies are seen. Creamy color colonies are especially yeast. Example, Cryptococcus neoformis, very nicely growing on the separodextrose agar after fungal culture, you have to see very nicely and creamy color. Yeast like fungi grow partly as yeast, partly as elongated cells resembling hyphae. Example Candida albicans. So, molds are filamentous fungi from true mycelia and reproduced by formation of spores. Example dermatophytes. Molds are filamentous fungi. Especially molds you have to see in filaments. You have to see in filaments. And dimorphic fungi can occur filamentous or yeast depending on growth conditions. In host tissues cultures are 37 degrees centigrade occurs is yeast and soils and cultures. Okay, these are four types of classification of fungi: yeast, yeast-like fungi, molds, dimorphic fungi. So, first of all, we have to study the next uh, fungus. First fungi is a Candida. Introduction: Candida albicans is a normal inhabitant gastrointestinal tract or oral and vaginal cavities. Uh, Microorganisms consist of fungi also. One type of fungi also microorganisms. Some fungi means especially Candida, they are inhabited. ఎక్కడ ఉంటుంది అంటే దాని యొక్క స్థానం గ్యాస్ట్రోఇంటెస్టైనల్ ట్రాక్ట్ GIT and oral and vaginal cavity. Oral means mouth. You have to see in mouth the Candida albicans presence in the mouth. Third one, vaginal cavities. Vaginal cavities. Vaginal cavities presence in the Candida also. Morphology. Hyphae show distinct points of constriction resembling sausage links. Budding yeast forms blastospores. Candida is an oval or spherical budding cell. Candida is a oval or spherical budding cell. Produces pseudomycelia in culture and host tissues. They produce opportunistic infection. Candida is a causes for opportunistic infection. Mostly, especially in diabetic patients. Opportunistic infections in diabetic patients. Lesions of skin and mucous membrane, superficial over thrush characterized by development of white patches or buccal mucous membrane. Candida albicans lesions, skin and mucous membrane. Superficial oral thrush and characterized by Candida albicans, and vaginal thrush also, vulva, vagina and cervix. In males, in penis involved, skin the moist area like axilla, groin, submammary folds are infected with Candida. Nails also, Candida is a causes for nails also. Nails, Candida infects toes and fingernails in persons. Especially Candida affected from the humans, nails, toes and fingers. So, cultural characteristics, generally their growth in Candida 37 degrees centigrade and sabradextrose or SDA. How to detect Candida albicans you have to see in germ tube test. So germ tube test is positive. Creepy colonies are seen in several dextrose agar. In laboratory diagnosis, uh, laboratory diagnosis you have to in candida you have to see in germ tube test. Second one is uh, chlamydospores develop on meal agar and 28 degrees centigrade. Second organism, Cryptococcus neoformis. Continuation tomorrow. Thank you all of you.